morning, students. Welcome you all. Today we are going to learn the basics of lipid biochemistry, and it will include the definition of the lipids, general classification of lipids, and the biological significance of lipids. After watching this video, I hope you would like, share, and subscribe my channel, Veti Biochemy. Thank you. So let's go. So our topic is the biochemistry of lipids. Oh yeah, it's a marker here. And the topic is the classification and biological significance of lipids. Before going to the classification, we will define what the lipids are. See, uh, lipids, they are the biologically organic molecules that are diverse, structurally heterogeneous, hydrophobic or amphiphilic, small water insoluble compounds extracted from obviously living organisms by weakly polar or non-polar solvents like chloroform, acetone, or ether, benzene, etc. So in this example here I have shown you that the upper portion, yellow color portion, this is the fat or oil, or I should say lipid. So you can see the two contrasting phases here. One is yellow and another is almost transparent. The lower one, this is the water, and the upper one, this is a lipid portion. You can say oil. So both of the two phases are immiscible, and that is why this group of molecules, lipids, they have been classified only on the basis of their physical property, that is the insolubility in the polar solvents. So the lipids are the substances which are immiscible in polar solvents, and it includes a diverse, structurally heterogeneous, hydrophobic organic molecules. And what is a distinguishing feature, property of the lipids? Uh, since I told you it's a diverse, heterogeneous group of the molecules, and you will see there is there is a structural diversity, a lot of structural diversity among these lipid molecules. You will not see there the homogeneity like in carbohydrates, like in amino acids, and what do we call it, is nucleotides. You will not see that type of similarity in the structural similarity in lipids. They are structurally different, very different. One lipid shows a significant structural difference with respect to the other lipid. But the property which keep all these molecules in the same group, that's a lipid group, is their insolubility in the polar solvent. So the definition, the lipid, this definition is based on the physical property, that is the immiscibility or the insolubility in the polar solvents, in contrast to the definition of the other building blocks. As I told you, that the proteins, carbohydrates, and nucleic acids, their 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 classification is based upon the structural similarity, their basic units, monomers. So we distinguish these like proteins, carbohydrates, or nucleic acids on the basis of their chemical structures. But in case of lipids, they are structurally diverse, different from one another. So which property keep them in the same group? That is their physical property, not the chemical property, not the chemical structure, their physical property. And what is that physical property? That is their immiscibility or insolubility in the polar solvents. One more point, lipids, they are not made of a monomeric units. Like carbohydrates, they are made up of monosaccharides. Their basic unit is a monosaccharide. Like proteins, their basic unit is amino acid, and it is the polymerization of these amino acids or monosaccharides. They make the macromolecules like carbohydrates, proteins. Nucleic acids, their fundamental unit is nucleotide, and when the nucleotides undergo polymerization, they made nucleic acids like RNA, DNA. But in case of lipids, you will not see there any monomeric unit, isn't it? So this is one more distinguishing characteristic feature of the lipids with respect to other macromolecules. And if you want to have advanced knowledge related to the lipids, for research purposes, for the new classification, which you can get the latest information about the lipids by clicking on the given websites here. So we will go to the classification of lipids. Before that, let's talk about the biological significance of lipids. Very important point is this. What are the biological, what the functions or the biological, really the what is the basic function of these lipids in the living organism? Here we will talk about, in general, we will talk about the animal tissue. What is the biological significance of lipids in animal tissue or in animals? So first of all, we all know that the lipids serve as a energy depot, isn't it? In the body, they provide us energy. They serve as a storage form of energy. So since lipids are of two types here, I should say, one is a storage type and another is a structural type. Structural in the cell membrane structure, we can say, and the storage type which we can store for the energy purpose, like triglycerides. So lipids have a major role in energy supply. They provide us energy twice as compared to the carbohydrates. This is the first time, this importance of biological significance of lipid. And one more important is that they serve as a signaling molecules, like eicosanoids are there, phosphatidyl inositol derivatives are there, thromboxanes are there. They serve as a signaling molecules. They carry signals from the extracellular environment to the intracellular environment. This is one more function of the lipids. Yeah, I told you that the dietary fats what we take while having a diet, we take actually fats for providing us the energy, because their main function is they provide us the high energy as compared to carbohydrates or proteins. And fats, they have also protective function in insects, in plants, and almost invertebrates. They serve as a part of the outer coating between the body of the organism and the environment. So they have a protective function here. And fats, like lipids, they are used for the synthesis of specialized signaling molecules. I told you that the prostaglandins, thromboxanes, and leukotrienes, they are all produced from the lipids, and they serve as a signaling molecules. Now, one more important function is that uh, biological lipids serve as a cofactor, like vitamin K. They serve as a hormones, like vitamin D. They serve as a detergents, like bile salts, which are produced from these liver and excreted through the bile. Few examples I have quoted here, like cholic acid, deoxycholic acid, chenodeoxycholic acid, lithocholic acid. These are all the bile acids, and when they are converted to bile salts after reaction with the alkalis or salts, they have a detergent action. They cause emulsification. They cause emulsification of the fats, therefore assist in the fat digestion. Uh, See here, I have shown you the example here. This is the actually the adipocyte. Adipocytes, that means the cells which are meant for the storage of fats. They are mostly found in the in under the skin as subcut tissues. They are found in the these cells contain high concentration of triglycerides. These are the lipids. We will discuss these triglycerides in next class. But the fat containing tissues are called as adipose tissues in the body, and the concentration of these adipocytes is high in mammary glands, in abdomen, in thighs, and they are rich in triglycerides. And these triglycerides are called neutral fats because they don't have any charge. Here, this is an example. Here, this is the adipocyte. Here is a nucleus, and this yellowish mass. This is the fat reservoir, or This is the lipid concentration, lipids that are stored here. These are the lipids. This yellow color mass, it is a lipid mass there. There's these triglycerides, and these triglycerides, when they are broken down by lipase enzymes, they are degraded, and then these fatty acids, they are hydrolyzed. These triglycerides are hydrolyzed, and the fatty acids are then subjected to the beta oxidation, and they release energy. uh, what is the one more function of these lipids is that they assist in the rapid conduction, neuroconductions, what we call saltatory. It is not salutary, it is saltatory, sorry, it is the saltatory, T-O-R-Y, saltatory conduction due to the non-polar lipids. They assist, they provide the sheath around the myelin, these non-polar lipids act as electrical insulators and allowing rapid propagation of depolarization waves along the myelinated nerves. Okay, and they also serve as a cushion around certain delicate organs like kidney, like eyeball. Beneath the eyeball, on the rear side of the eyeball, you can see there's a fat deposition of very much fat, which serve as a cushion for the eye. Okay, so they have a cushion like function. And these lipids, when they are attached with some certain proteins, what we call the lipoproteins, they together serve as a lipid carrier in the blood. Okay, and uh, they serve as a means of transporting lipids in the blood. And these lipoproteins also serve as a structural component of plasma membrane, cell membrane, and mitochondrial membrane. And fat, I told you that the fat which is under the one more function is that the fat which is under the skin, that is a subcut fat, or which is called a subcutaneous tissue, which is rich in the adipocytes. It functions as energy store, as I already told, as well as the insulator against the cold. So it is a very important function of this. uh, so it is very important to be to have a knowledge of the lipids. And there there are so many other functions of these lipids in the biological system, but these are the general functions, and I believe every student must know these functions or the significance of the lipids in the living organism. So here I have put some examples of these bile acids. This is the glycocholic acid, taurocholic acid. This is the bile acids. These are the lipids which assist for the deposit, for the digestion of the fats by emulsification of the fats. Okay. The knowledge of lipid biochemistry is necessary in understanding many important biomedical areas. Okay, now let's talk about the classification of lipids. Simply, lipids are mainly categorized in the three general categories: simple lipids, compound lipids, and derived lipids. Okay, simple lipids are those which are made by esterification of fatty acids with the alcohols. Okay, and the compound lipids are those which are formed by the esterification of fatty acids with the different groups of alcohols, and fatty acids. Fine. So compound lipids are the esters of fatty acids with different groups of alcohols and fatty acids. Now, derived lipids. Derived lipids are those which are formed from the hydrolysis of the compound or simple lipids. So let's talk about the simple lipids. Simple lipids, these have two main groups, fats and waxes. Fats, they are the esters of fatty acids with glycerol. So here alcohol is the glycerol, and examples of these fats are oils and neutral fats. Neutral fats and oils. It has two examples I will show you in the next slide. And now the waxes, the second class is the waxes. These are the esters of fatty acids like in the fats, but Here, the alcohol is not a glycerol, it is a high molecular weight alcohol. Fine? A long chain alcohol is there. They together make a waxes, and waxes are solid at room temperature. Fats can be solid or liquid. Fats can be solid or liquid depending upon the degree of unsaturation. We will discuss in the later. And now, if we talk, let's talk about the compound lipids. What are the types of the compound lipids? It includes five groups. First is a phospholipid. Phospholipids are lipids containing, in addition to fatty acids, they contain alcohol, phosphoric acid residue. Fine. They frequently have nitrogen containing bases and other substituents like in glycerophospholipid, the alcohol is glycerol, and in sphingophospholipid, the alcohol is sphingosine. Okay, but if we talk about the glycolipids, they are the lipids which include uh, fatty acids, amino alcohol like sphingosine, and carbohydrates. Okay, we were discussing the glycolipids and sulfolipids. Welcome back to the channel. And sulfolipids, they are the fatty acid and made up of fatty acids, alcohols, phosphoric acids, and sulfur containing groups. uh, Sulfates, usually they contain sulfates as additional groups. That is why we call them as sulfolipids. And then aminolipids, definitely the aminolipids contain fatty acids, alcohol, and amino acid. And regarding these lipoproteins, I told you that the lipoproteins, mm, they are actually the complex of lipids and proteins together, and they have very clinical significance related to the disease developments, especially cardiac. Derived lipid, they are the, I told you that they are derived from the hydrolysis of the simple and compound lipids, and it usually contains the fatty acids and cholesterol on hydrolysis. And fatty acids may be saturated or unsaturated depending upon the presence or absence of double bond. If the double bond is present, then they are called as unsaturated fatty acids, and if the double bond is not present, then they are saturated fatty acids. Here I have put some examples here. See, if we talk about the simple here, see the simple lipids, they contain triglycerides and waxes. Triglycerides means the glycerol is esterified with three fatty acids, and the fatty acids may be saturated. If all the three fatty acids in the are attached with the glycerol and they are same, then that is known as simple fatty acid or simple triglyceride, like tripalmitin, tristerin. Okay, and if they are saturated, that means no double bond is present in the fatty acid. And fatty acids, if they contain double bonds, then they are called unsaturated fatty acids, and if they contain one single bond, they are known as monounsaturated fatty acids. The best example of this MUFA, or the monounsaturated fatty acid, is oleic acid, and polyunsaturated fatty acids are those fatty acids which contain two or more than two double bonds. The example of this polyunsaturated fatty acid is linoleic acid and linolenic acid. Now, the example of phospholipids, lecithins, phthalins, we will discuss these one by one in next classes. And glycolipids, I told you last time, it includes cerebrosides, gangliosides, and in derived lipids, you have steroids, bile acids, sex hormones. Under sterols, you have a cholesterol, ergosterol, and other carotenoids, or we can say that they are chromolipids. These carotenoids are chromo, they are color containing pigments like carotenes and xanthophylls. Okay, now simple lipids. We have two examples of simple lipids. There's a fats and waxes, and fats are the esters of fatty acids with glycerol. If we and fatty acids here, the fats uh, esters or the fatty acids and glycerol. These are the fats. Triglycerides, diglycerides, monoglycerides are the fats. In fact, if the glycerol is attached with one fatty acid, then it is known as a monoglyceride. If two fatty acids, then diglyceride, and if three fatty acids, triglyceride. Let's see the diagram. You will get a more clear concept regarding these. Huh? See here, it's a glycerol and is a trihydric alcohol, and if three fatty acids are esterified here with this glycerol, see the glycerol part, and you have this ester bond between fatty acid and alcohol. This is if it is only one, like here mono is known as monoglyceride. If the two fatty acids are esterified with glycerol, then it's diglyceride. If three fatty acids are esterified here, then it's a triglyceride. Fine. Now waxes, they are the solid fats, simple fats. Then the waxes are defined as esters of fatty acids with high molecular weight alcohols. Like we have a good example of wool lanolin, lanolin wax or bee waxes. They are well known, and these are actually formed by the see it's an example here, bwax is an ester of the fatty acid palmitic acid and an alcohol, that is triacontanol alcohol. It's a 30 carbon alcohol, very high molecular weight alcohol. And let's talk about the biological significance of these waxes. And here waxes, they form the protective layer over the skin, fur, feathers of animals, and give a shiny appearance to fruits. Fine, and see, this is a protective layer in the sense that they they form a protective layer in the sense that uh, they they make a barrier between the harsh external conditions and between the tissue. So so to save the animal or the save the tissue, they have a, that is why we call them, they have as a protective layer. They have a protective function, these waxes. And of course they are very hard at low temperature and soft like gel like, become jelly like when at high temperature. And lanolin, we all know that it is the animal origin is obtained from the wool of the sheeps, and it imparts the sort of insulation, and it prevents the loss of heat from the body. Now let's talk about the fatty acids, because the fatty acid is a component of simple lipids. It is a component of these compound lipids, and when these simple and compound lipids undergo hydrolysis, they produce uh, they produce fatty acids. Fatty acids, these are the simplest lipids. Okay, these are the simple lipids and can be oxidized to generate the energy needed by the cell by fasting. In the fasting state, excluding brain and erythrocytes. These cells, they obtain their energy from the glucose, not from the fats. Fatty acids are the precursors in the synthesis of more complex cellular lipids like triacylglycerol. And let's talk about the essential and non-essential fatty acids. If the fatty acids can, if a fatty acid can only be obtained from the diet for humans, I'm talking about, then the fatty acid is an essential fatty acids. But if we will see in ruminants, they have almost their rumen microflora, they manufacture all the fatty acids. That there is no essential fatty acids in ruminants. But in case of monogastric animals like we, animals or dog or cat, we do need these essential fatty acids uh, from the external sources, from the external feedstuffs. Okay, if I would talk about this essential fatty acids, uh, I told you that if a fatty acid can only be obtained from the diet, then the fatty acid is essential fatty acids. And the best two fatty acids that cannot be synthesized, best example of these essential fatty acids is a linoleic acid and linolenic acid. Fine, these two, linolenic acid and linolenic, in case of humans, we have these two essential fatty acids. These are essential fatty acids for our body, because our body cannot synthesize these two fatty acids. And one thing is that both the fatty acids, linoleic acid and linolenic acid, they are unsaturated fatty acids. Both are unsaturated fatty acids. Now, if we will talk about non-essential fatty acids, these fatty acids can be made by human body, and so do not need to be obtained from the diet alone. Okay, these are made from the carbohydrates and proteins or from other fatty acids. Fatty acids are an important source of energy. You remember this word, they are the important source of energy, because finally these fatty acids are broken down into the acyl groups, and these acyl groups are then are diverted towards the Krebs cycle for the production of energy. And we must know that the carbohydrates and proteins, they provide only about 4 kilocalories of energy per gram per gram, while as if we talk about the fatty acids, they provide almost 9 kilocalories per gram. So these fatty acids produce the energy more than the double. And this is one of the reasons why a high fat diet can lead to obesity, huh? Because of deposition of calories. And thanks for watching this video. We will meet soon with a new topic.